All right, just a bit about the vestibular system. This system is about balance, and you hear me talk about the vestibular apparatus and the central vestibular nucleus being primarily involved in this. Now, the vestibular apparatus is within the inner ear and it detects motion, position, and it transduces this information to a neurosignal um, where then it is uh, taken to the central nervous system. So then we're talking about the vestibular nuclei, which are primarily concerned with maintaining the position of the head when in space. And the tracts that descend from these nuclei mediate mostly head on neck and head on body adjustments. And so when we talk of the vestibular apparatus, in summary, what we're going to be talking about is angular acceleration, horizontal linear acceleration, vertical linear acceleration, and decelerations of all of this. So we'll talk about semicircular canals when we talk about angle, and then we talk about linear acceleration. If we're talking about horizontal, side to side, we'll be talking about the utricle, and up and down, we're talking about the sacu. All right. So anatomically, the vestibular labyrinth is the one that contains the structures that respond to balance, and the system has the bony structure and the membrane structure. So we're talking about two systems that work together. So the vestibule is a bony structure, okay? It's a bony structure that has the membrane structure, and the membrane structures are the sacs and the utricles that I've just talked about. Now, the sacs and the utricles have sensory epithelium that are then used for detection of balance. Another component that is attached to this is the semicircular canal, and that it has ducts and epithelial sensors known as cristae apularis. This is usually for kinetic energy. So if we look at the diagram that we have here, I said what we have is a vestibule that is a bony, okay, it's a bony um, structure, and then it has membrane structures that are known as the utricle, okay, utricle, and the sacu. So they have utricle and sacu. So not only do we have that, but then we also have some canals. So these canals, we they are semicircular, and at the bottom of each of these we have cristae ambularis, and the cristae are the one that hold detection apparatus of whatever fluid is um, in these canals. So we have cristae at the bottom of each of these. So why do we have them at an angle? So that all angles or angular movements are detected. So we have a superior angle, okay? Then we have a lateral angle, okay? And we also have a posterior. Having said that, the vestibular apparatus is concerned with this ear detection of motion and position and transduces these signals. So starting with the sacs, and the utricle. What we want to say is these are found on the floor. They're found on the floor of the bony part. And they use maculae as the sensory components. So they also have support cells, and you will hear me make mention to our hair cells. So when you talk about the utricle, you're talking about horizontal movement. Okay, so horizontal, but what type? Not angular. We're talking about linear. Okay, this way and that way. So it could be acceleration or deceleration. They are able to detect that. And when we talk about the sacu, we are talking about them also having very similar detections, but for these we say that they are all about the vertical. So it's vertical but still linear, and we're still talking about acceleration and deceleration. So the sacu and the utricles, we say they use the macula. So what is the macula? What are we really talking about? So the macula is this, okay? When you talk about the macula, we're talking about an autolytic bone-like organ, and it is at the floor of both the utricle and the sacu. Okay, so at the walls of both of these we find the macula, and we see that they're able to detect different linear accelerations because of the way that they are placed or surmounted on the walls. So these, if I could just clean up, you will find that they have cells, and these cells have within them the hair cells. Okay, they have hair cells, and these hair cells have similar types of hairs that we're talking about. Hearing. Okay, now on top of these hair cells, you find that they have surmounting them this autolytic membrane, and this autolytic membrane is that's named because right on top of it are the structures that I'm drawing, and they are known as, someone to guess, autolyths. So these autolytic membranes, between them and the hair, you have this gelatinous matrix. Okay, so of course these embed into this membrane, but in between them you have this gelatinous type of matrix that is there. And then of course we have our axons that are coming out. Other things that you should know is that these are all about crystals of calcium carbonates. All right, so it's all about calcium carbonates, and they can also be referred to as autoconia. It's more dense than endolymph, and the process of the hairs, these ones, okay, are embedded into the membrane. So if the membrane moves, then we find that there's movement. If it moves this way, it means that there's tilting this side. So when we talk about linear acceleration, we see that autolytics will move to the back and cause action potentials that then are taken to the central nervous system. So in the utricle, so we said the utricle is all about horizontal. So if it moves this way, actually I've done it the other way, but the principle is the same. If it moves this way, you've seen that the hairs are aligned like that. So if it moves this way, again we're talking about tip lengths. We'll find that there's increase in depolarization and hence action potentials. Okay, so on our drawing, we had them right at the base. Okay, we had them right at the base, so we had them there. So movement can be horizontal, but whichever way that they move, we see that these help stretch the tip length, causing an action potential to occur. The opposite is true if we're going to have hyperpolarization. So this movement this way will cause hyperpolarization. The saccus are placed in this way. Okay? So movement is up and down. Okay? So for the vertical, going up, okay, so these are just tilted to come and be this way. Right, so going up is going to tilt our hair cells this way, and once they're tilted that way, you're going to have action potentials. And then sliding down, of course, will allow an alteration. So again, in the vertical, the going up will slide the autoconia, okay, to slide the autoconia, and you'll find action potentials eminent, while the opposite is going to happen, allowing the autolytic to go up, and the pattern of action potentials is altered. So again, if it is this way, if you are going to, if there's a gelatinous arrangement there, and then you have your tip links, okay, what will happen is, sorry, I keep on drawing them the other way around. Okay, so if you have your tip links this way. When you're going up, this, because it's heavy, is going to slide down. Okay, so because it's heavy, it's going to slide this way. Once it slides this way, it's going to carry these that way. Okay, so this is the opposite of what I had done here. So it's just the opposite. Okay, again, we have our otoconia, which when you start going up, they go down. Because they've gone down, tip links will be carried down. And once that happens, you'll find that the tip links will be stretched. Because tip links have been stretched, you have an increase in action potentials. Opposites will lead to hyperpolarization. Okay, so that's what's happening with the autolyts on the sarces and utricles. What about the semicircular canals? The semicircular canals, sensory epithelium is known as cristae. So you are looking at okay, the sensory epithelium is the cristae, and it's on each side of the head. And the semicircular canals are perpendicular to each other, so meaning at every angle of movement, every angular movement, you find that one of the semicircular canals is really, having its fluid move and stimulating the cristae itself. So the crista ampullaris is located in the expanded end of the, as a receptor, as we noted in the diagram earlier. So the expanded end is the ampullaris. So if we could just go to this drawing, we have the ampullaris, okay? And then we have the cristae itself. Okay, and what we have here is a membrane, and in this membrane then we have the hair cells. Okay, I always have to remember that the right side is the tall side. Anyway, depending on which side of the ear you're talking about. So the cristae, this is the crista ampullaris. This itself, as I said, is this one is the copula. We have supporting cells, we have hair cells, and here we have endolins. The crista have hairs and support structures, and then they're surrounded by a gelatinous partition. Okay, there's this gelatinous partition that closes off the ampulla. Okay. So another thing that you might want to know right now is that the process of hairs is embedded in the copula. So this part in the copula and the base of the hairs are close to in contact with the afferent fibers. So you see that the copula gets deformed in a direction opposite to that during acceleration. So if you're moving this way, you'll find that the copula moves that way. And once it moves that way, it's going to then pull these tip links, and we have an action potential ensuing. So most of the copula goes in one direction, increasing firing of this single fiber. If the opposite occurs, okay, so movement this other way, you find that the copula moves this side, and then you have an increase in hyperpolarization. Okay. So as part of your homework, just want to know which side of the ear has what type. Okay, which side of the ear has what type, and where do the hairs, where is the cilia? Okay, so is it this way and is it that way? Is this your left and is this your right? Let's use our anatomy to study our physiology. Okay, so if it's this way, it means that when you turn this way, this turns that way. So which one of these shoots up action potentials? All right. Read up as well on the notes that I've given you on vertigo, Meniere disease, as well as motion sickness. The central pathway has also been labeled in your notes for you, so go through them. Ensure that you note that the cell bodies of the crest of the macula are located in the vestibular ganglion. Okay, so let's just clean that up. So these are in the vestibular ganglion, and from the vestibular ganglion we have nerves that terminate ipsilaterally, and this ipsilateral termination is you will notice that there's a 4-part vestibular nucleus that you're talking about. Then fibers from the semicircular canal end primarily in the part known as the superior and the medial divisions of this, and then they project to control the eye. Whilst fibers from the utricle and sacu usually are noted to, fibers from the utricle and sacu end predominantly on the lateral component. So these project the spinal cord as well as the cerebellum. So after this, you have an involvement of the thalamus. Fibers project to the thalamus, and from the thalamus, they then go to the primary somatosensory cortex. So ensure that these are studied. So let's just go back to what I have given you as what to study. You have the right side and you have the left side. If you have your copula, you also have your copula, left side, and your nose there. All I'm saying is, what is the direction of your hair cells? Okay? Are they in this form? And if they're in this form, if you move this way, or rather, let's move that way. If we move that way, actually we'll leave that there. It means that you're moving the copula this side and this side. So you're going to have action potentials and and here you're going to have hyperpolarization. So to detect that movement is from here to there. So let's study. So is it like this, or is it this way, and then the other side is also this way? Okay, we'll discuss this at the beginning of next term in class. Have a good vacation.